Hi there, and welcome to my booth. Just to start off with a bit of introduction here, prostate cancer is the most commonly diagnosed cancer among Canadian men. It is a predominantly androgen-dependent disease, relies on androgen receptor inhibitors such as enzalutamide for treatment. The problem here lies in the fact that many patients develop resistance to these inhibitors. And one mechanism by which this occurs is referred to as lineage plasticity or neuroendocrine transdifferentiation, in which these cells are initially of an adenocarcinoma cell type will transdifferentiate into these neuroendocrine-like cells that no longer require AR for growth and are molecularly characterized by loss of RB and P53 expression. What's actually quite commonly seen in clinical biopsies are these mixed tumors, suggesting that these cells may transition through a stem-like cell intermediate state. However, the molecular mechanism of how this occurs is not well defined. But there has been one genomic study that identified RB as the most significant factor in determining poor survival for enzalutamide-resistant prostate cancer patients. So that brings me to my hypothesis, which is that mutations in RB misregulate stemness and cellular differentiation pathways that result in an increased propensity to transdifferentiate and acquire enzalutamide resistance in prostate cancer. So to test this hypothesis, I first performed a genome-wide CRISPR knockout screen in a known prostate cancer cell line LenCap cells to identify gene mutations that confer either increased resistance or increased sensitivity under enzalutamide treatment conditions. To also further understand the functional aspect of RB and P53 loss in prostate cancer, I used CRISPR-Cas9 to generate RB P53 double knockout LenCap cells, and then performed various enzalutamide resistance assays with them, such as an acute 6-day enzalutamide treatment to measure IC50 values, as well as a chronic 4-week treatment to allow these cells to form colonies over a longer period of time. I also performed RNA sequencing on both double knockout and LenCap parental cells. Volcano plot in figure 1 here is showing genes that were either positively or negatively selected from the CRISPR screen. And one gene that I want to highlight here is the stem cell factor HOXA9 that was negatively selected in this screen, suggesting that the function of this gene is important for LenCap cell survival under enzalutamide treatment conditions. And as expected, we see that both RB and P53 score as positively enriched, again confirming their role in promoting enzalutamide resistance. Now, when we look at RB-P53 deficiency, surprisingly what we find that over an acute 6-day enzalutamide treatment, there was actually no difference in IC50 values between double knockout cells and LenCap parental cells. However, when these cells were treated with enzalutamide over a chronic period of time, only the double knockout cells were able to form distinct colonies. Furthermore, RNA sequencing of double knockout and LenCap parental cells showed that neuronal processes and as well as development and cellular differentiation pathways were significantly upregulated in double knockout cells compared to LenCap parental cells. And again, we see the stem cell factor HOXA9 as significantly upregulated in double knockout cells. So the fact that it's scored in both our CRISPR screen and RNA sequencing data led us to want to investigate this gene further. Now, interestingly, we found that HOXA9 expression levels are positively correlated with neuroendocrine features in enzalutamide-resistant prostate cancer patients, and also negatively correlated with RB expression. So to understand the functional aspects of HOXA9, we use CRISPR-Cas9 to mutate or to induce mutations in HOXA9 that led to its overexpression at both the mRNA and protein levels in both the LenCap and double knockout background. And surprisingly, what we found is that cells that overexpress HOXA9 were highly resistant to enzalutamide, as shown by increased IC50 values over an acute 6-day treatment, as well as significantly more colonies, as well as larger colonies formed over a chronic 4-week enzalutamide treatment. So what these data suggest is that RBP53 deficiency can alter gene expression programs that promote lineage plasticity and enzalutamide resistance. Furthermore, our data suggests that the transcription factor HOXA9 is important for LenCap cell growth, and that its expression can cause cells to become highly resistant to enzalutamide. We believe that HOXA9 may be inducing a stem cell-like intermediate state that promotes enzalutamide resistance. So overall, this suggests that HOXA9 may be a potential biomarker and therapeutic target for the treatment of enzalutamide-resistant prostate cancer patients. Thank you.